Everyone. Welcome, everyone. I'm Tony Latai, president of the Society of Functional Precision Medicine, and I'm glad you all could join us here today. This is the sort of the last seminar of the season until we have a break. I want to remind everyone there will not be a seminar in August. Then the seminar series will take up again in September. Also, if you are a member and you think you have something interesting to present, we are filling our 2020 for especially the latter half, but we are filling our 2025 calendar. So please contact me or Aaron directly. You can see options for contacts on the website too, but you can certainly feel welcome to email me if you have a talk that you'd like to give and would like to be considered. I'd like to remind everyone of a couple of sort of business items. One is we have just recently accepted nominations, including for board positions, including two new board positions for people representing commercial interests. Very good response to that. You will be getting notified about how to vote. And if you are a member, you will be getting information about how to vote in this election in your email. I encourage you to vote. It's a sign of a healthy organization to have plenty of voting for these positions. Of course, when you run for a position, some people get elected and some people don't. If you don't, I encourage you to get involved. These not only these board positions, but in general, there's a lot of work that the SFPM does that is probably very relevant to your business. Including, for instance, we have a regulatory working group that's I think, been doing some very excellent work. And there's other things that I think that our commercial members will bring to us. Please keep your eyes open for opportunities to contribute to that, because that's really how the work of this SFPM gets done. We don't have a big administrative staff taking care of things. It's you, the members, who do these. I also want to remind everyone of this meeting in Copenhagen that we are co-sponsoring with the European Hematology Association at the end of September. Details are directly on the front page of our website. I encourage, if you haven't already, to register and bring people from your laboratory. This is a great opportunity to demonstrate the strength of our field. Part of it is attending meetings like this. We were encouraged that something that the SFPM could offer its membership is to start holding meetings. So that's what we're doing. And I encourage everyone who is listening to this to consider attending. Should be a great meeting, also in a very fun location. I also want to mention there is another meeting that we're going to be advertising on our homepage that SFPM is not sponsoring, but we definitely are in favor of, and there will be SFPM members like Diana Azam speaking at it. And this is the PMNet Forum that's going to be hosted 10 to 11 of October in Riga, Latvia. I went to that meeting before, and it was a great meeting and really interesting to see Riga as well. So I encourage you to keep your eyes open for that. I think there's a vibrant series of meetings involving functional precision medicine in the next year, and I hope members of the SFPM will participate at many different levels. So at this point, I think I will introduce today's speaker. Today's speaker is Ali Jonas. He is an associate professor of radiology at Brigham and Women's Hospital and Harvard Medical School. His research mainly focuses on medical device development, including interests in biomedical optics, spatial systems biology, and precision oncology. Of course, it's the latter that's going to interest us the most for the sake of this society. He is also a professional quality hockey goalie, although I don't know, he doesn't advertise that as clearly in his CV, but I just happen to know that from before. So he's going to present today on an implantable microdevice platform that he's developed in his lab that can measure drug responses in patients with solid tumors. And I'm very excited about this, not only because I've been able to watch the development for several years, but because they're actually doing clinical trials, and that is extremely valuable to the progress of functional precision medicines to perform these sorts of clinical trials. So I think it'll probably work best if you hold your questions till the end. You can write them in the chat, or you can raise your hand at the end of the talk. So without further ado, I'll let Ali, please everyone mute your microphone unless you are Oliver Jonas, and I'll let Ali take it away.
Okay, sounds great. Thank you, Tony, for the introduction, and thanks for having me here. It's great to meet everyone and tell you about our work. I'm okay with interruptions along the way. If there's any sort of technical questions that can be helpful if they're answered right away, before we move on with other slides, please feel free to either put the questions in the chat or raise your hand or interrupt in some other way. So the work I'd like to tell you about is our implantable microdevice platform. And it's, I think as Tony has alluded to already, a functional precision medicine approach, but it's I think in many ways different from other approaches, and I think it's very complementary to many of the other approaches that people are working on and have developed, as well as complementary to other precision medicine approaches like the genomic and other techniques. So our technology is really based on sorry, first my disclosures. Our technology, like others in the field, is really based on the idea that a lot of current therapy selection in oncology is still empirical, that a lot of it is based on standard of care that sort of applies broadly to many patients that fit certain criteria, and that it often proceeds from a first line to second line, third line, and that the goal of precision medicine, of course, is to be able to distinguish between potential efficacy of multiple different treatments and to choose the one right off the bat that will end up working the best. In oncology, I think that's especially pressing, where you have so many different drugs available that have very different mechanisms of action. These drugs are very expensive, and of course, I think we all believe that an optimized drug selection or combination selection can improve survival and reduce side effects and potentially lead to cost savings for the system as a whole as well. And especially on the combination side, I think there are very few techniques now that are available to really sort of rationally select treatments. And I think with all the excitement and all the incredible progress that's been made on the genomic side, I think there are still true that the vast majority of cancer patients do not actually benefit from a genomically guided treatment selection. And so I think the case for functional precision medicine in general is still very much intact and may even be growing as time goes by and more treatments and more combinations are being considered. So the approach that we wanted to take was to essentially to use an engineering approach where we make measurements directly in the patient's tumor. And the main reason for that is, of course, that there are many factors that contribute to tumor drug responses. It's not just the genome. There's also all the other things that are happening that contribute to the phenotype. And so we wanted to make those measurements on the phenotype. And there was a concept that the FDA had published over a decade ago now that was called the phase 0 microdosing concept. And the idea, I think, originally was more on the PK side, to be able to enable PK studies of drug distribution and to do those using very small amounts of drug. The definition at the time was less than 1% of what would be considered a traditional systemic dose. So essentially administer microdoses systemically, and then you can learn about how those drugs distribute, potentially how they're metabolized in the body and so on, without actually having to expose a patient to systemic and potentially toxic systemic doses. Our idea was to take that microdosing approach, which has by itself a favorable regulatory pathway, but instead of using it for PK measurements, actually use it for PD measurements. So to create small regions in the body that are exposed to therapeutic levels of drugs, where we can actually measure PD effects, but to do that in only a very small part of the body, so that overall it would remain a microdose, and then you could eliminate these systemic toxicities that you may encounter from therapeutic systemic doses. And so this is the approach that came out of that. It's a small drug delivery implant that can be placed directly in the tumor. It has many different reservoirs on it. Each reservoir has a different drug, and allows you then to measure many drug responses in the tumor at the same time. So this is a little more detail on the technical workflow, and there's a little animation to go with that. So there are some exceptions to this clinically, but for most tumors, the procedure is as follows. We use a biopsy needle to place the device, and it's usually a 19 or 20 gauge needle, and then the device remains in the tumor for usually about 3 to 5 days. And during that time, we have different drugs in different reservoirs. There's 20 reservoirs per device. It can also be combinations. It doesn't have to be single agents. And then after those 3 to 5 days, the tumor with the device in the middle is resected, and then it's processed, and then we thin section that tumor device specimen. And this is also shown on the left here. And you get these different sort of zones of drug diffusion for each individual agent, and they're spatially separate. And you can examine the response of the tumor to each of the drugs individually and in parallel. So this again can be up to 20 different agents. We've used a wide range of different sort of therapeutic modalities: small molecule cytotoxics, targeted agents, antibodies, antibody drug conjugates, combinations between different types of drugs. And those we've all used clinically already. In the animal setting, we've used other compounds as well, like nanoparticles or oligos, for example. So there's a lot of flexibility in terms of what can be delivered with this approach and studied in the tumor in terms of different types of therapies. Any questions at this point? Okay. So in terms of what the platform can do and how it can be used, so I mentioned that each of the devices has 20 different reservoirs. We can always place multiple devices in the same tumor, and we do this routinely clinically. It's normally about 3 to 4, but in some cases can be 5 devices or more. And so you can actually, if you have different drugs on different devices, you can study up to 100 different drugs or combinations in the same tumor. In a lot of tumor types, we don't have that many. So a typical set of drugs is around 15 to 20 different therapies that are tested. It's at a microdose, or actually much smaller than what the FDA considers a microdose. We sometimes call it a nanodose. So they're very small amounts of drugs, but they're delivered in such a small part of the body that it ends up being a systemic microdose, even though locally it's at the therapeutic level. And this lets us measure the concentration of the drug locally, and then the effect of the drug on the tumor. So essentially create PK/PD curves. I'll talk more about the details of that later. But this combination of measuring drug concentration across a whole range of concentrations and then drug effect, I think, is really important. And because this is happening directly in the patient's tumor, it sort of takes into account all the factors that contribute, all the cell types that are present in the tumor. So I think it's a great way to look at immunotherapy responses, for example, at combinations, and so on, metabolite levels, and I'll show examples of those later. And of course, combinations. And in terms of predictive biomarkers, the idea is to find essentially markers that correlate with systemic drug response, and then in the future, hopefully use those to inform clinical decision-making as to whether a given drug should be prioritized for an individual patient. In terms of what are potential applications for this, I wanted to put this slide a little bit earlier so that people can kind of think about that as we go through the data. I think first and foremost, it's the personalized therapy selection. I think that's what most of us are interested in. Taking the sort of pool of available treatments, identifying treatments that are likely to work in a patient, and then to actually give that. That would likely be mostly approved treatments, or potentially novel combinations. And then that sort of leads to the second application, which we think is in developing new drugs. So being able to use this platform, not necessarily to say, okay, what drug are we going to give this patient systemically, but to see if we can get early clinical data novel compounds, on investigational compounds, on phase 1 compounds, for example, to see how that compound acts on the tumor. What are the biomarkers of response and resistance? Can we understand in which patients this drug works and in which it doesn't, so that we can develop a given drug more effectively? And that can be done both in the preclinical setting in animal models, but also clinically as part of sort of a phase 0 study. And then the third application is one that I'm very excited about, and that's the kind of big data component. Is we're able to make a tremendous amount of measurements now from within a single patient. Can we connect those drug response measurements with all the other data that we're collecting from the patient's genotype data, for example, and other clinical data, and can that lead to a better understanding of of drug responses in general for given tumor types? So the first part I want to start with is how do we actually measure tumor drug responses with this approach? And the first part of that is, of course, the release of the drug, and that is has two components. The first one is how do we control the drug, and the second one is how do we measure it in the tissue. So in terms of the control, so all of the drugs in a given reservoir are formulated in a polymer, and the polymer controls how much of the drug is released, how fast it's released, and to make sure it does not release too far in the tissue, or it may overlap with others, with other agents, etc. So this polymer formulation is really important, and it gives us a lot of control over that local drug concentration, and it allows us to tune those local concentrations to what is achieved from systemic dosing, for example, or to any other desired concentration that one wants to measure in the tumor. So this is an example here from a study using doxorubicin, where we administered that systemically, and then we measured in the tumor what the actual drug levels are, and then we tuned the concentration of the formulation in the device so that it recapitulates that same concentration range. One thing that's important is that the release from the microdevice is always in a spatial gradient. So when you're close to the reservoir, there's a higher concentration. When you get farther away, it gets lower and lower. And so this is kind of that PK/PD component where you can measure drug responses across a range of concentrations because you have that natural concentration gradient that's distance dependent. And then the second part is the measurement of tumor response. So I mentioned that there's a concentration gradient. So closer to the device, you have higher concentrations, and as you get further away, you have lower concentrations. So we then spatially overlay that drug gradient with measurements of effect, and this is a sort of simplest possible measurement here. This is a single antibody measured by IHC for cleaved caspase-3, so looking at whether a given drug induces apoptosis, and then overlaying this with the concentration distance dependence, then allows you to get these kind of PK/PD curves. What percentage of cells are you killing in response to a certain concentration? We're starting to look at how many of these different markers we may need to get a predictive value. This is a study that was done a little while ago in animal models, in ovarian PDX models, where we ended up finding that we probably need about 5 to 6 different features, different markers that actually give us a very high predictive value. And then there are many more markers that we can use for exploratory measurements, for biomarker discovery, etc. And that's really a big part of the effort now. And this is just an overview of 3 techniques that we use a lot now, but this is not an exhaustive list. There are basically any types of kind of spatial biology measurements that you can make on an FFPE slide or on a fresh frozen slide. Can also be used, but these are 3 examples that we use a lot. So the first one is cyclical immunofluorescence. This is a technique developed by Peter Sorgo and others. And it allows us to measure about 25 to 30 different antibodies. And you can use that then to characterize cell types in the treatment zone, and of course cell states like apoptosis, proliferation, etc. So this is an example here from a head and neck cancer patient where we released pembrolizumab, and we're able to profile that sort of localized immune response very precisely with CISIF. And then two other examples of spatial techniques that we use is the spatial transcriptomics. We use the nanostring platform, but there are several others out there now that really provide an incredible amount of data from even a single treatment region like the one shown on the left here. You can do a whole transcriptome analysis, and you can do that for all 20 drugs individually on the device and compare them to the control regions nearby, or compare different drugs to each other. And then the last example of a technique that we use on some of our samples is MALDI mass spectrometry to do spatial metabolomics, so that you're able to measure metabolite levels and how they change in the tumor in response to a localized drug treatment. And there's an example of adenine here for one of our brain tumor samples. So that was sort of a basic techniques approach. How do we release the drugs? How do we control them? How do we measure the response in the tumor? The next thing I want to talk about is, I think, the one that's very important, maybe the most important aspect, is how do we use those local readouts to connect those to systemic treatment responses and ultimately to clinical responses and clinical outcomes. So we've done a number of studies in animals and now in patients that look at that question. The first one was a study shown here, which is in triple negative breast cancer tumor xenografts in mice. And what we did there is we tested some of the cytotoxic agents that are used in that disease. We tested them locally with the device. We tested them systemically. And for this small set that was examined here, we saw a very nice sort of one-to-one correlation between what works locally and what works systemically. More recently, we've looked at immunotherapies and immunotherapy combinations. And this is using the cyclical immunofluorescence approach that I described on the previous slide. And this is an example of what that matrix of markers looks like. So you have your, a lot of the markers are used to essentially phenotype your cells. So your myeloid cells, your lymphoid cells, and then look at things like immunogenic cell death, and then, cell state like apoptosis, proliferation, etc. So it's a manageable set of markers. It's still a fair amount of work to conduct that, that cyclical staining, but there are other techniques now where this is becoming more automated, and also where you can do a lot of protein markers like this in a single shot, like CyTOF, for example, or others. But it's essentially you're trying to get the same type of data, phenotype the cells, and then identify responses both on the tumor cells and on the immune compartment. So that's what we did here. And we took a number of targeted agents. This is an example here for palbociclib, CDK4/6 inhibitor. And what we found is that when we deliver these agents locally, we see very specific and characteristic drug responses in the tumor that involve both the tumor cells and the immune cells that are present in the tumor. And for each of those targeted agents, for that specific drug response signature, we were able to see how that drug fails, for example, or can potentially fail. So for palbociclib, for example, we saw these pro-tumorigenic macrophages that are emerging in the treatment zone, and that's generally associated with a poor outcome. And so the rationale derived from this was to combine the palbociclib with an anti-CSF1R antibody. And when we did that, we saw a vastly superior response, both to the compared to the agents individually, as well as compared to other combinations. And then we went through that exercise for multiple different agents, and for each of them, you get these specific response phenotypes, and then a direct, sort of combination rationale from that. And some of those examples are shown here. And then we went a little bit sort of into the mechanism of why that could be, et cetera. But essentially, it's a purely agnostic approach where you just deliver the drug, you measure what the tumor does, and based on what you observe, you then choose a combination agent. And we showed that that gives a superior response. We always place multiple devices in the same tumor. I think that's an important part in terms of tumor heterogeneity, of course, to be able to to get multiple replicates for the same drug from the same patient. But also, I think it gives us a little bit more than that, in that we're able to better understand how different factors in the tumor microenvironment may affect drug response. And the advantage here is that because the devices have a very precisely loaded drug and formulated drug, and a very precise location where it's being released, we're not subject to the sort of PK variability that you get from systemic dosing. And so if you see responses in one region and not in another, and you have the same drug release profile, it really lets you take a closer look at the biological differences and understand sort of potential mechanisms of biological resistance to certain drugs much better. So even though we often think of tumor heterogeneity as sort of an impediment to studying drug responses, I think in this case, there's also an opportunity to study it more precisely and to actually use it to understand the role of the tumor microenvironment in drug resistance more precisely. I have a couple examples I wanted to mention of how we've used this in the research setting, and then wanted to transition to the clinical setting. So we can also study sort of biological compounds and even biological probes with this. It doesn't have to be drugs. It's a little sort of kind of a technical demonstration here, where we want to study metabolism directly in the tumor, using metabolic probes that you can administer systemically. So this was a paper where we showed that macropinocytosis is a mechanism of sort of nutrient scavenging in pancreatic tumors, and we released this fluorescent albumin drug conjugate locally in the tumor, and we're able to show that this protein catabolism is only happening in the tumor cells, and not in the other cells in the pancreas. And then secondly, we wanted to look at the mechanism, and so we used this one compound called EIPA, which inhibits macropinocytosis, which is the mechanism for taking up sort of large proteins into cells, so they can be metabolized. And when we release that compound locally, we're able to block the metabolism specifically. So doing both kind of the hypothesis, like generating the hypothesis and then also confirming it using the same approach. Another example is we wanted to study tumor metastasis, and especially cell migration in the context of tumor metastasis formation. And so there were these breast tumors that have a splicing variant called MENA invasive, and the hypothesis was that MENA invasive reorganizes the collagen network in tumors and can use those then to preferentially migrate along those gradients. And so we tested that by releasing fibronectin directly in the tumor using the microdevices, and to create those gradients ourselves in the tumor. And we're able to see that the MENA invasive subtype was the one that actually ended up migrating along those fibronectin gradients, whereas the other subtypes were not. So again, testing a biological hypothesis by using a biological compound rather than a drug to study. But again, doing that directly in the tumor, I think it enabled this approach, doing it locally rather in a systemic study. And the last example is one, again, looking at metabolism, but taking advantage of the fact that you're able to induce very strong localized phenotypes, same as what I showed there from the Nature Biotech paper. It's often very difficult to study tumor metabolism on a single cell basis in vivo because you have sometimes, for example, T cells that are sparse, and your sort of spot sizes on the laser for MALDI or for other sample types. You can't really study single cells. It's more clusters of cells, and so you don't tend to get very strong signals if there's one T cell that's surrounded by many tumor cells, for example. It's tough to say, okay, well, what's the T cell metabolism contributing to the overall sum of those cells? And so being able to create these really strong phenotypes locally has a big advantage because you get aggregates of cells that are much more uniform. And so what we're able to do here is to study both the tumor cells and the immune cells, which we're characterizing by SciSif, and then to overlay that with the MALDI, which is shown on the right, and that gives us specific metabolite levels, so we can see which metabolites are up and which are down. And you kind of go through that kind of step by step, and you see which regions are high in certain metabolites and which low, and then it gives us sort of a first look in vivo of how different competing cell types adjust their metabolisms, basically. So if there are any questions, happy to stop for a moment. Otherwise, we can move into the clinical work. So it's obviously in the whole microdevice approach is very different from what has been done to date, using different approaches from a technical perspective. I would say even if the ultimate rationale is essentially the same for most functional precision medicine approaches, but we have a device, we've got drugs, we've got lots of different drugs. They're formulated in different ways. All of that goes into the patient. It has to be taken back out, etc. So there are lots of regulatory hurdles that we had to overcome. And a lot of it sort of had to be newly developed because there wasn't a clear precedent. So what we ended up developing with FDA and with the IRBs that we're working with is a process that allows us to develop a clinical protocol that describes all the procedures, all the kind of formulation of the drug, how the device is stored, how it's sterilized, etc. And that goes into the protocol with the relevant appendices. The FDA reviews that first. Sometimes it's an iterative process. We get comments, we make changes, etc. Eventually, the FDA approves the IND, and then the protocol goes to the institutional IRB. There's again comments, etc. And then once approved, the study can proceed. We now have 10 INDs at the Brigham and Dana-Farber, where the microdevice is being used in different cancer types. And in conjunction with this process on the left, there's a separate process. There's essentially a whole quality system that we put in place that details every step along the way, from how the devices are fabricated, the traceability of the material that's being used, preparing the microdevices for sterility and endotoxin removal, loading the drugs, and then testing sterility and endotoxin, which is done externally, and then packaging devices, labeling them appropriately, and then delivering them to the research pharmacy, which holds them until the day of the procedure when they're implanted. And that whole process, even though it might sound a little complicated, I think in practice it's worked quite well, and is now being translated to other institutions. We now have trials that are outside of the
Ollie, can I ask a question?
Sure,
This is
yeah.
the one of the challenges that we have found in moving forward is the need not only for an IND if you're testing novel combinations or something like that, but also the need for a separate regulatory pathway, an ID, investigational device exemption. And you literally have a device, even though
Yeah.
they consider ex vivo diagnostics also to be a device. That's why I mentioned it. How do you get around the requirement for an IDE?
So it's not technically that we're getting around it, but the FDA has, and this happened with the very first protocol that we submitted, and they followed the same pathway since, that they look at it as a combination product, a drug device combination product, but that they consider the drug side to be the more important side, I guess, because the drugs are the thing that's acting on the tumor rather than the device, which is supposed to be inert. And so it's still a combination product. There are reviewers on the device side, but the lead is on the drug side. And so what they issue in the end is an IND. But I think part of the reason for that, again, is that I think the terminology was the primary mode of action of this combination product is the drugs. And so they consider that to be the lead. And so they're issuing INDs.
Let me ask it differently. Have you been in clinical trials where you have used this device to make therapeutic decisions for a patient, like put them on one arm or another?
Not to date, but that's the goal for the future.
Do you expect
not?
It might be, maybe that's the difference. It might be that when you get to that point, they will require an IDE of you. What do you think?
I think that's totally possible, yeah. And certainly when it gets towards as you said, like the actual clinical decision making, potentially a market approval, et cetera, I think at that point, yes.
Yeah, that totally makes sense. I think that the IDE, so the IDE, which for anyone who's not familiar with it, is very focused on the performance, the analytic performance of whatever your diagnostic is. However, that is most significant when you're actually going to use it to make a decision based on if you're not using it, I can see why that would escape that. And I can see why the IND would be what was really required.
Yep. So I'm looking forward to going through that process in the future.
If I might add a comment, it's Pam Becker. I just wanted to mention that just like your IND can be deemed that you're exempt, I had that the IDE was also exempt. So I went through the process, but my assay for acute leukemia, they exempted from IDE as well. So just like they exempted the IND and they also exempted the IDE.
How did they exempt it, Pam? Were you using it to make decisions?
Yes.
So how on earth would they exempt it?
It's being used to make decisions on a clinical trial. So it was investigational, and they think that I can go back and try to go through the review and why the exemption. I mean, the IND was exempt also. I think that the IND was exempt because I wasn't trying to license anything, and I think that's an important point. You know, my assay, I wasn't trying to license it or anything, and then I was. I think they said that because of the extremely poor prognosis of the patients, that they think that's why what led them to exempt they weren't eligible for any approved therapy, or had already failed all approved therapies. So they were quite, I guess, I mean, I felt like they were incredibly supportive of this, these new innovations. And so at that time, a few years ago, when I had submitted the AML, the acute leukemia multi-omic approach. So I just wanted to mention that it could be their decision to exempt it. And I'm not sure, we were CLIA approved, and I don't know if that helped, but they granted us the IDE exemption. I can go back and try to read the reviewers' comments on everything to try to answer this question better for the group and follow up with you. But my impression was that it was similar to the IND exemption. So I just wanted to let everybody know that, it takes a huge effort, as far as I understand, it takes a huge effort and very costly to try to get an IDE approved. I mean,
I can definitely
effort.
vouch for the accuracy of that statement.
So we were
Pam,
fortunate.
I think that we talked about this when you had presented your awesome data at ASH. I think that your study is about a decade old though, right? I was wondering whether or not the
No,
FDA had changed their stance, because we have a similar high risk AML no available therapy study, and they want both. So it's been a bit of an uphill battle.
they,
Uh,
do they want more than the submission? I submitted for IDE. I'm just wondering, do they want IDE?
Validation. They want a validation. IND won't proceed until the IDE is approved. So it's been evaluated by both agencies.
I see. So
Yeah.
it's not, yeah, it was, this wasn't my very first trial, which is, my first trial was older than my more recent trial. This was my more recent trial. So this wasn't the one that was the first trial. So this was like, I'd have to go back to find out the exact dates, but maybe 6 years ago, I think. So not 10 years, maybe 6 years ago. But and was your assay CLIA approved? Is your assay CLIA approved?
No, what we were told is that the CLIA approval wasn't really relevant, and what we needed was an IDE.
Yeah,
Mhm.
I can add to that. So I didn't have a CLIA approved test, and I went through exactly what Pam went through, and we got determination that this is non-significant risk because all patients had no other curative options. So we had to include that in the informed consent, that or the criteria that there are no other curative options or they had very poor prognosis. So and I got an exemption as well.
Yeah,
Okay,
we had requested that and gave the same rationale since it's the same disease, and they said it was not acceptable.
I want to let Ali get back to the, I think one learning here is that there can be great variety in the FDA decision-making when they're on the cusp of their knowledge base.
Maybe just one comment or question on that real quick. It sounds like maybe if if there are multiple options for patients, or if you're trying to change them away from standard of care, that would be considered a risk. But if you don't have those options and it's considered like less than minimal risk or minimal risk in that regard, that maybe you do get a favorable pathway, then I'm not sure if that makes sense. But I'd love to hear more from other people about that offline as well, if possible. So we've used the microdevice now in 10 different cancer types. There's a couple new ones here, bladder, for example, that's not yet on the graphic, but over 190 devices implanted, 75 patients to date. Very well tolerated, no serious adverse events related to the device. We've looked in a subset of patients if we can even measure any of the drugs systemically using mass spec and other sort of ultra-sensitive techniques, and we can't. So the drug remains local, which is important also for showing kind of a safety, safety of drug exposure outside of the tumor, and then wide range of drugs tested, many of the available cancer drug modalities. We haven't done cell therapy yet, but that's something we're working on in animals, so that might be a way to test that as well, CAR-Ts and others in the future, but that hasn't been done in patients yet. In terms of clinical implementation, so I wanted to talk a little bit about how the device is placed and how that procedure works. It's usually an image-guided biopsy placement. So you can see the device here, an example of it here, shown inside the biopsy needle. And we use either CT or MRI or ultrasound to sort of see the needle advance into the tumor and then deploy the device directly at the tip of the needle. This is very similar to a sort of fiducial placement or a brachyseed or similar type and size devices that interventional radiology routinely places in patients. So we've had extremely high success rate here, and think the procedure is very sort of routine and safe as well, or at least as safe as a normal biopsy would be. For device retrieval, we typically, at this stage at least, we're placing devices before surgery. So if a patient is scheduled for surgery, we then implant the device 3 days before, for example. And then in terms of the measurements and kind of the tissue processing, it's usually very fast, and so you're able to get data or the first data at least, you know, a few days after the tumor's taken out. Some of the more high-plex spatial transcriptomics, small d, et cetera, would take longer, but some of the faster readouts like the IHC and even some of the cyclical staining can be done within a few days. And so you could conceivably incorporate some of those results at some point in the future into the clinical decision making, which I think is really important too. This is an example I wanted to show from our GBM trial. So obviously not the easiest site to place and retrieve devices, but it's worked remarkably well. And the surgical team, Dr. Peruzzi, at the Brigham, has done an incredible job there. So this is an example of a recent patient where the patient is scheduled for surgery, and then 3 days before surgery, there's a small biopsy that's performed here. This is actually indicated, so that it's done to confirm that it's a glioblastoma in this case. And then at the end of that biopsy procedure, we just leave the device, in this case, 2 devices here, 1 and 2, in the tumor, and you can see the device here on a small sort of forceps there as they're being inserted. And then we put a little bit of tissue glue on the top so that it stays nicely in place. And then, it's closed up for the duration of that interval between the procedure and the surgery. And then the patient comes back 3 days later. We confirm that the devices are still in the tumor, haven't moved or migrated. And then the patient goes on through the standard surgery. And then this block of tissue here, you can see containing the 2 devices is resected. And then that's the specimen that we receive in the lab, and then it's processed the same way I described earlier, where we section through that laterally, and then get cross sections of the different drugs on the different levels of the device. This is a different example of a clinical implementation in lung cancer patients. So some were done, robotic surgery, some were done open thoracotomy, and devices placed again through the needle, and then retrieved at the time of surgery, and then just examples of different measurements and comparisons between drugs. But sort of, I would say, 2 of the more difficult anatomical sites, and devices can be placed and retrieved there quite well. This is a technical approach that we're working on to make the devices usable without surgery. We already do that in skin tumors, but for other lesions, deeper tumors in the body, we developed a biopsy tool that goes directly over the microdevice and retrieves tissue only in the region where the zones of drug release are. And we think that this is a good way to sort of place it and then keep it in for 3 days and then come in with a larger biopsy and retrieve it. So this is first being tested in breast tumors in patients, and will then be used in other superficial tumors, and then hopefully in the longer term in deeper lesions as well. So I think this will be an important aspect of using the devices in, for example, metastatic disease or other indications where the patients are not going to surgery. So what have we learned from some of the early measurements so far? So we had a pilot study in brain tumor patients that was completed. The first look was, of course, safety feasibility. That was, I think, very favorable. Slightly longer surgery, but not a major difference. Cost was the same. ICU stay, total hospital stay. So well integrated into the clinical workflow, and technically feasible in terms of placement and retrieval. Some of the patients in the study received temozolomide systemically after the surgery in the adjuvant setting. And so we wanted to look at whether the measurements that the microdevice made at the time of surgery were predictive of the systemic response that the patients had when they went on systemic treatment after the surgery. And so we had these 3 patients here. They were, interestingly, the methylation status was not a good predictor of response. I think it's generally considered to be unreliable, especially for partially methylated MGMT status and for unmethylated. But in this case, we had a patient with a partial methylation status who had a very good response clinically. And that was also the patient that had by far the strongest response on the microdevice. So one response that was unexpected by methylation status was shown to be strong on the microdevice and ended up being a very good response clinically, both in terms of the sort of tumor shrinkage, but then also survival. And then patients 5 and 6 here had no response on the device, or very, very low response, and no response clinically resistant to temozolomide. Actually, one of them was methylated, so would have been predicted to have some response, but didn't, and the device predicted that correctly. And another patient that was unmethylated didn't have a response clinically, and the device also showed no response. So very small cohort, but I think encouraging early data. And the study is now being expanded to more patients, and we actually just opened a second site at UCSF to help with enrollment. So we're hoping to get sufficient numbers there over the next year or two to really look at that question of can the device accurately predict responses in a statistically significant way. In addition to the sort of diagnostic aspect, if we want to call it that, there's also the biomarker discovery aspect. And so in addition to temozolomide, we had a lot of other drugs on the devices in the brain tumor patients, and we wanted to do some biomarker discovery and some kind of target validation, target engagement type studies. And so we looked at some of the targeted agents that were on the device and wanted to confirm, or see if they're hitting the target, for example, and see what else, what other effects those agents induce. And so we showed for these different agents that they're actually hitting the target that they're supposed to for abemaciclib, cell cycle checkpoints, for everolimus, mTOR, for osimertinib, EGFR, for lapatinib, EGFR and HER2. So you can pull that nicely out of the spatial transcriptomic data. On the metabolism side, we found some interesting responses, and one of those is shown here for lapatinib. So we have the exact drug response, the drug release zone of lapatinib, which is shown measured here. And in that release zone, you see some very definitive biomarker signatures. Here, this is an example of glutathione being highly upregulated in the treatment zone. And then we identified 2 new metabolites here that were not previously known that are upregulated in the treatment zone, and are looking at metabolic signatures in sort of an unbiased way. So from a biomarker discovery perspective, I think this is really neat because you're taking advantage of that localized release. You can compare that release zone with the directly adjacent untreated tumor, and it really lets you kind of find what's up and what's down in response to the drug treatment. And then the sort of last example is one related to the question of big data. So we have a lot of different modalities we can measure. How do we put that together? How can we take advantage of the high precision of drug exposure and measurement, and then take the different modalities and learn more from them together than we would individually? So this is an example of that is we're working with a partner who's interested in sort of H&E, machine learning, mostly based on H&E samples. And so we're trying to understand and really train sort of larger models as to how those H&E profiles of tumors change under treatment. And in a normal clinical trial, you're only able to test one treatment at a time per patient, or one combination at a time. And with our approach, you have 20 times that. And so we think that we could grow these data sets much more quickly to to really, you know, get to that point where the models are trained sufficiently so that you can draw inferences on tumor drug responses from those sort of common and high throughput techniques like H&E pretty quickly. So this is an example of kind of combining the different measurements that we do, like the H&E and the SciSif, and then the spatial transcriptomics to be able to do that single cell registration and then overlay of the different measurements. This is another example of just the vast density of data. So looking at the pathways that are up and down regulated. I mean, you can from every single treatment zone, you're getting hundreds of pathways. You can see how they change in response to drugs. Have them as single agent, in this case, nivolumab alone versus nivolumab and olaparib together. So what does the single agent do? What does the combination do? How can that overcome potential resistance mechanisms? How can it activate favorable pathways, et cetera? So just a tremendous sort of depth and amount of data from each local measurement. And then to do that in an unbiased way, using either machine learning or a kind of principal component and other statistical tools to see, okay, I know I have drug exposure in this region here. You know, how is this tumor region different than all the surrounding tumor regions? And then what are the metabolites that are up and down regulated? So kind of a computational kind of, I guess, biomarker discovery approach that we're pursuing here. And the last thing I want to mention is that even though we've talked almost exclusively about cancer, we think this technique can also be used in other diseases. So this is an example from Alzheimer's model where we place this directly into the brain, and then we're able to make measurements in a way similar to what's done in the oncology models or in the cancer patients, but in some cases using different readouts, different measurements that are more relevant to those diseases, and then of course using different drugs on the devices, but the same technical approach. So with that, I just want to thank everyone for listening, and then also thank the people in the lab, especially Sharath and Christine, who've done a lot of the work that I showed today, and then the rest of the lab and the funding that supported this. So if we have any time for questions, we'd be happy to take those.
Great, thanks, Ali. While I'm looking for hands to go up or questions in the chat, I have a question, which is that it seems to me that one of your primary measurements made is of cleaved caspase 3, which many people use as a measurement of apoptosis after some perturbation. Seems reasonable to me. So I'll ask you a question I always get asked. What about drugs that what do you think, how many of these drugs are not inducing apoptosis? And do you run the risk of overlooking drug effect by focusing merely on apoptosis?
Yeah, I think you're entirely correct on that. I think there is that risk. And so there may be different measurements that are needed for different drugs. And I mean, if you have a target inhibitor, I think you would also want to see what the drug does to that pathway, right? I mean, so I think the answer is yes. I think there are going to be different markers needed for different drugs, but I also think there is a common set, for example, for the cytotoxic drugs that you're using. I mean, if you're comparing 5 cytotoxic drugs, then I think markers like cleaved caspase 3, Ki67, cleaved PARP, some of DNA damage markers like phospho-H2AX, some of those. I think a small set of those, and my hunch right now would be a set of, I would say 4 to 6 markers is probably going to give us a very good idea, and more comprehensively too, as to which drug would be more effective. And then for immunotherapies, you're going to have different markers too. Like you'd want to see T cell activation, you'd want to see maybe CD8 as one of your readouts, maybe granzyme B, maybe CD11C if you have antigen presenting cells, et cetera. Like those would be relevant markers for those drugs. And I think the ultimate answer is we don't know yet which markers will be needed for different drugs, but I think we're getting a better understanding already. And that's definitely one of the goals for the future, is to define those readout sets more precisely.
Great. We have a question in the chat from Sean Mahoney, who is interested in the estimated time of arrival of your trial coming live, and is it going to be open to adults only or open as well to pediatrics?
So all the trials that are open now are only for adults. We are talking to the pediatric group here. So there may be a trial in pediatric patients coming up in the near future, but it's not live yet.
Okay, as everyone knows, both in drug as well as diagnostic development, pediatrics always lags behind, generally because of smaller patient size and other logistical aspects. They always do seem to come last, which is a major problem of its own. I think we have reached, in fact, gone a little past the top of the hour. Thanks for a great talk, Ali. It's always great to get a refresher on the exciting work that you're doing. And is there a place where people who are interested in these, say, just the clinical trials, is there a place where they can just refer to and see where they and like a link or something you could provide?
Yeah, definitely. They're all listed on clinicaltrials.gov, so I can provide some of those numbers, might be helpful. And then if there's any other information, happy to share that with the group later.
If you wanted to email me and Aaron those numbers, we could post them up on the SFPM crawl, and people could see them there.
Sure, yeah, sounds good. I'll do that.
Okay, great. Thanks. All right, thank you, everyone. Have a nice August, and we'll see you back in September. That concludes this seminar.